Boa tarde a todos para mais um seminário da pós-graduação do IB e da FM, já que a gente inclui programas das duas unidades da Universidade de Brasília. E eu queria agradecer muito, já de antemão, a presença do Dr. Addison Lana, que gentilmente aceitou dar esse seminário sobre esse tema tão importante para a gente, né? E lembrar vocês todos que estão entrando aqui pela primeira vez, que ainda não fizeram, que, por favor, subscrevam o nosso canal, ativem o sininho, que são as notificações. Tudo isso, gente, ajuda a gente a subsistir no YouTube. Então, eu queria deixar com vocês com o professor Guilherme, que é do programa de Patologia Molecular. Tá? O professor Guilherme vai apresentar o Dr. Addison, e desde já eu agradeço muito ao Guilherme e ao Addison por toparem estar nessa atividade com a gente, tá bom? A palestra será em inglês, o Dr. Addison é americano, mas não se esqueçam que vocês podem fazer a pergunta, inclusive em português, que a gente vai fazer para o Dr. Addison ao final da palestra dele. Então, Guilherme, por favor. Thank you very much, Addison, for staying with us today. Okay, Guilherme, please.
Olá, pessoal. Boa tarde. Bom, é uma grande alegria aqui e agradecer primeiramente aí a organização da André, dos programas de pós-graduação. É uma excelente iniciativa. E, obviamente, agradecer ao Addison. Addison, I'm going to translate afterwards, just a few words in Portuguese. E realmente é uma grande alegria ter o Addison, que ele veio lá de San Diego para passar 2 meses aqui no meu laboratório. E a gente se encontrou lá no laboratório do professor Chris Glass, que é uma sumidade aí na área de imunologia, de nanomacrófagos, com grande potencial de aplicação genômica. Eu, na época que eu cheguei lá, eu fiz um pós-doc lá em 2019 para 2020, e aí no meio teve essa pandemia, mas mesmo assim a gente conseguiu trabalhar bastante. Foi um grande desafio para mim começar a entender a genômica. Eu venho o meu background é mais estudando a parte estrutural de proteínas e DNA, e lá eu mergulhei dentro dessa área genômica. Eu acho que é o que o Addison vai abordar agora. Bom, o Addison, como eu falei, ele estava já no laboratório do professor Chris Glass há aproximadamente 3 anos e pouquinho, quase 4 anos já, na verdade, 3 anos e tanto. E lá a gente se encontrou e ficou grande amigo. Uma das questões que fortaleceu bastante nossa amizade é que ele também é um lutador de jiu-jitsu e é um surfista, e alto nível nos dois. E também é um grande cientista, como vocês vão ver o trabalho dele aí, que acabou de recém-publicado aí na Science, que ele está envolvido em um grande grupo aí, e é o que ele vai apresentar para a gente. O Addison, ele fez biologia e seguiu diretamente aí para pesquisa no laboratório do Chris Glass, e agora ele está entrando aí na escola de medicina e continuando seus estudos. E se tudo der certo, aí eu ainda vou conseguir segurar o Addison aí por mais um tempinho aqui no Brasil. Addison, so I'm just explaining how we met each other, yeah. And of course, I think you understood that I also said that one of the things that we got the stronger relationship over there is that also you are a jiu-jitsu fighter and a great surfer. So besides to be a great scientist, I think you have a more diversity in terms of interest in what I think is really cool and can open and increase the creativity. So Addison, I will not extend. I just pass to you. Thank you very much, and this is a great pleasure for me to have you here and to have you here in my lab, and also on the mats. Okay, so please, Addison, with you.
Yeah, absolutely. I'll get started. Hey everyone, so my name's Addison. First off, let me share my screen real quick so you can see my presentation. Should be good. Okay. Also, please let me know if anyone has questions during, because I really want to, I know there's a lot of people, but I would like to have a discussion as well. If there's any questions, stop me, feel free, please. Because it'll be better than waiting until the very end if you have any questions, and I can hopefully go through some things, because there's some complex methodologies involved in what we do, and that I definitely didn't understand before I started working in the class lab, and now I have a better understanding of, and I hope I can share some of our expertise and knowledge of what we do with you guys today. I'm super excited to be here and talking to you guys. So I'll go ahead and get started. So hey, again, everyone, my name is Addison Lana, Javier Lana. Today I'll be presenting on some work that Johannes Schlahetsky, who is a assistant project scientist in the glass lab, Hunter Bennett, who is a recently graduated MD PhD, so he finished his PhD portion recently, and myself have done over the last couple of years with the help of many other members in the glass lab and Philip Gortz's lab at UCSD. And this is on using the epigenetic and transcriptomic profiles of microglia in MPSIIIA. So MPSIIIA is mucopolysaccharidosis subtype 3A. It's a lysosomal storage disorder, but I'll get into that later. So using the microglia, the profiles of these cells in this disease setting to study other neurodegenerative disorders. So as I said, I worked in the glass lab. It was the Department of Cellular and Molecular Medicine at UCSD. Let's see if I can't. Okay. So first off, what I'll be discussing is the importance of lysosomes as metabolic hubs of the cell. We've all learned about lysosomes in our basic biology courses, but I think that they kind of go underappreciated sometimes. And I definitely underappreciated them before I started doing work on them. And we want to look at how they're involved in both controlling and processing both intracellular supply as well as extracellularly obtained nutrients, and through recycling intercellularly pre-existing macromolecules through a process called autophagy. And this will help in a starvation setting where you can use pre-existing nutrients that you have or your cell has to be able to compensate for a lack of nutrients, as well as, what our data suggests is like there's a heightened lysosomal function in a microglial activation context. So we want to know how a possible impairment of lysosomes can function or lead to microglial activation, or what the connection is if it's the microglial activation leading to lysosomal dysfunction or vice versa. And as well, which later we'll be discussing, is the role of MITF or TFE transcription factor family members in causing inflammation, not only within microglia, but in most cells in this disease context. And we'll use this model to study other neurodegenerative diseases. And that is mainly the reason because a lot of other models of, for example, Alzheimer's disease, Parkinson's disease, Huntington's disease, they're, as much as they are genetic diseases, they also are aging. At least Alzheimer's diseases is very involved with aging, and it takes a long time to see the manifestation of the symptoms in the context of Alzheimer's disease. And the current models we have, for example, different mouse models that have expression of amyloid beta, hyperphosphorylated tau, things like that, they show symptoms and manifestations of the disease, but they're not necessarily modeling the exact disease processes or modeling exactly what's happening in this disease. And in this case, in MPSIIIA, which I'll get right into, I'll discuss briefly right now, is a dysfunction of a specific enzyme leading to an accumulation of glucosaminoglycans in lysosomes, resulting in lysosomal dysfunction, overactive autophagic processes, as well as phagocytic processes in immune cells, leading to potential neuroinflammation and neurodegeneration. So we're able to know exactly what's going on. It's a very rapid model, and we can figure out how to treat this and use this to study other diseases. So lysosomes are crucial for cellular homeostasis as well. And their activity is tightly modulated by the presence of different nutrient levels, such as glucose nucleotide levels, as they do recycle nucleotides and amino acids as well. And this is in part regulated by autophagy, which we'll talk about again later, as far as recycling in nutrient deficient states of things that are already existing, pre-existing within the cell, and involved with recycling those macromolecular subunits. And they're also involved with cell growth, as they're controlled in part by signaling from growth factors and insulin response and things like that. And they're also involved with cell growth. So in the context of cancers or mTOR, which will be relevant later, I keep saying that, but I have to preview these things so that you'll be aware of them once they come up, is that in these rapidly dividing contexts of cells, we see overactive lysosomes and an accumulation of these lysosomes, so division. We also see an immune response as these phagocytic processes are taking in different pathogens, different bacteria, things that are involved with immune response, as well as then processing like immunogen formation from antigens and things like that, as well as lipid metabolism. And these organelles are actually very sensitive to perturbation. So as I was talking about starvation, but what that also means is that we are better able to affect lysosomal function because they're so sensitive. So we're able to modify and affect lysosomes to try and understand what goes on in the cell in an environment where there's a disease happening that affects the lysosome. So we can mimic that or model that or affect the lysosomes in a certain way. And some things like advantages and disadvantages to having overactive lysosomes, for example. So in the context of MPSIIIA, we do have increased lysosomal function, would be you have a clearance of, as you can see in the bottom left, you have a little figure here, clearance of intracellular storage materials. So in this case, you're having heparan sulfate, which is, like I said, this glucosaminoglycan that builds up in lysosomes as it's not been able to be broken down due to a defective sulfamidase enzyme. And as you have an accumulation of this, you need to have an overactive lysosome or dividing lysosomes to try and accommodate for this, because even though you're not able to process these heparan sulfates, and having more lysosomes wouldn't allow you to process more heparan sulfates, you still need to address the current demands of the cell. So that might mean breaking down lipids, breaking down other sugars, breaking down things that aberrant proteins, things that have been misfolded, things like that, that are going to be used in autophagic processes to recapitulate those macromolecular subunits into their original processes, or into new processes rather. So some dysfunctions to actually having the increased lysosomal. So this is, and actually having that might be seen as good, right? So in the context of neurodegenerative diseases or lysosomal storage diseases, you see that, and that could be, it can be a good thing, but it also induces excessive catabolism and the promotion of like hyperproliferation and growth, so such as cancer, as well as it can even be in the context of digesting necessary cellular processes, such as in neurons. So you'll see neuron death and things like that. So why are we exactly studying lysosomal storage disorders? Because it seems like a very niche disease, and I'm not sure, but I hope some of you have heard about them in the past, but I hadn't before I started working on them, honestly. And why they're so cool and why I've grown to love, love that they're so similar and so useful for our study is that it's very common. Lysosomal dysfunction is very common in aging and neurodegenerative diseases. So as your cells are overtaxed with aging and your cellular processes are growing in demand. So let's say that there is a context of being in Alzheimer's disease, Parkinson's disease, or Huntington's disease, you get aberrant protein aggregation. So in the context of Alzheimer's disease, you'd have amyloid beta or hyperphosphorylated tau, or alpha-synuclein in the context of Parkinson's disease, or Huntington protein aggregation in Huntington's disease. Now, what we can do is we can learn from lysosomal storage disorders, and that's because you actually see an accumulation of cholesterol, of hyperphosphorylated tau, of alpha-synuclein, of amyloid beta in these neurons and in these brains of patients, and in our mouse model, you see the similar manifestations of these neurodegenerative disorders in lysosomal storage disorders. So if we know exactly what's going on in these disorders as far as why this is happening, and we can study the processes as far as like how the cell's accommodating for these differences and dysfunctions, and how we can try and alleviate some of the overactive inflammatory responses and immune responses of the cell to accommodate for these problems that they're not equipped to handle. So if we can address those and then treat with some type of gene therapy or gene therapy or AAV mediated gene therapy or different types of protein therapies, we'll be able to maybe address other neurodegenerative diseases as well. So lysosomal storage disorders, as far as the one that we were interested in most, is MPSIIIA, as you can see from what I've circled here. All of these different lysosomal storage disorders, so MPS II, MPS I, MPS IIIC, these other ones. The only main difference, and yes, they do have slightly different manifestations, is the accumulation of different sizes or types of these glucosaminoglycans, or these either heparan sulfate, keratan sulfate, demetan sulfate. So specifically in what is called Sanfilippo syndrome, or MPS IIIA, you see a buildup of, like I was saying, heparan sulfates. And then not only does this affect the CNS, but it also affects the periphery as well. So we see it affecting nerves and the brain, nerves being in the CNS and periphery, the heart, kidney, spleen, liver, basically every vital organ that you can study in this context. Our lab has studied, not necessarily in the context of MPS IIIA, but it's possible to. So that's why we've been able to do the work that we've done, is we have the opportunity to work with other people that have specialized in different organs and immune cells in different organs to try and look at this in more in depth. So we'll talk a little more about the pathogenesis of this disease. So the accumulated mutation, specifically in the SGSH gene, coding for sulfamidase, a protein that breaks down heparan sulfates. You get an accumulation of these heparan sulfates in first the lysosomes, and then subsequently lipid and cholesterol accumulation as there is dysfunction of the lysosomes. And you'll see lysosomal overproliferation, as well as autophagic impairment, as they're not able to break down these lipids, these cholesterols, these sugars, these proteins to repurpose them. So you'll see accumulation of these aberrant proteins and faulty mitochondria as you're not able to break down mitochondria that's no longer functioning. And this will lead to, which we'll talk about, signaling abnormalities and ultimately immune activation when there really shouldn't be immune activation because there's not an infection. And inflammatory processes, immune abnormalities, and subsequently neurodegeneration, leading to cell death and organ damage. Now, this is a fairly rare disease, and it only affects 1 in every 70,000 births. And while it is rare, it's, like I was saying earlier, it's important to study. And that is because how similar these processes are in different neurodegenerative disorders. Right. So what is the underlying transcription factor network driving microglial states? So something that we should discuss first off is that transcription factors, which are these proteins that bind to either enhancer regions or like distal enhancer regions or near promoters, can actually affect gene expression. And that's how you get different cell identity, because these transcription factors will open up specific regions of chromatin, and this will only be present in specific cell types. So we'll get into it a little later, but in a disease context, sometimes cells will see an expression or manifestation of a specific family of transcription factors that in this case was MITF or TFE family members that drive autophagic specific gene expression, lysosomal gene expression, inflammatory gene expression, and might make the disease actually worse. So if we can address that by doing either some type of knockdown or treatment in some way to limit the accessibility of these different MITF and TFE transcription factor family members, then we might be able to address the gene expression that's leading to this damage-associated microglial state. So first off, neurodegenerative diseases are associated with multiple states of microglial activation. And these are called, in the field of microglia, they're called DAM, or damage-associated or disease-associated macrophages, or microglia. So lysosomes are genetic hotspots for neurodegenerative diseases. Basically, this is just saying that in the context of a lot of neurodegenerative diseases, you see an upregulation of lysosomal specific genes. And what our thought process is that is because that the autophagic processes, phagocytic processes, lysosomal processes are working on essentially overdrive to try and deal with some of these aberrant protein functions, protein aggregations. And the approach that we used was modeling MPSIIIA and Sanfilippo syndrome as a model system to study lysosomal dysfunction and subsequently neurodegeneration, and how microglia behave in the disease context and how they might be driving or affected by the disease. And the specific MPSIIIA mouse model, we had 2 different mouse models, and we used both to try and look at the differences between the 2. We first had a point mutation model in the SGSH gene, and the SGSH gene is located on chromosome 11. And this results in a reduced sulfamidase activity. Sulfamidase is still present, but it is reduced significantly. And then in the other, we had a deletion of exons 3 through 6 of the sulfamidase gene, resulting in a non-functional protein. And so this is kind of a pathway of disease progression. So let me drink some water. So you see a defective SGSH gene, inability to process, I'll use my pointer really quick. Inability to process the sulfated glucosaminoglycan heparan sulfate, leading to an accumulation of partially degraded heparan sulfate, and as well leading to leaky lysosomes and dysfunctional lysosomes. These processes are leading to, at the same time, activation of the innate immune response and inflammation via the TLR4 pathway in inflammasome, as well as endocytosis, phagocytosis, and antigen presentation impairment. It's affecting both of these pathways, and these are actually driving more dysfunction in and of themselves. They're going back and making this disease worse. These are all leading to rapid neurodegeneration. Microgliosis, meaning this damage-associated microglial state. So microglia normally are in their spindled form. They're pruning neurons. They're working together with different microglia. And then in their disease model, they don't have any branches, and they're expressing these very inflammatory genes and releasing cytokines and things like that. So that's what microgliosis is. And microgliosis also meaning that there's an overabundance of microglia present and neuroinflammation. These are all leading to, you'll see a buildup of cholesterol, gangliosides, alpha-synuclein, amyloid beta, and hyperphosphorylated tau within these cells. And so this is what I was discussing as far as the TLR4 pathway and inflammasome being affected. So you'll get heparan sulfate building up in these lysosomes, not able to properly be broken down as they're taken in, right? So then they're taken in here, SGSH is not working, but the defective or half digested heparan sulfates are expelled out of the cell, causing a reinduction of TLR4 activity and kind of this vicious cycle a little bit. And this also is leading to the activation of the NLRP3 inflammasome, leading to cleavage of caspase 1 and releasing IL-1 beta and things like that. So as I was discussing, this is some of our methodologies for how we isolated microglia in the disease context, mice as well as wild type mice. So I'll go back to discuss again this deletion of exons. This is one of the mouse models we have, is the deletion of exons 3 through 6 of the sulfamidase gene SGSH. And what you'd see is in the point mutation as well as in this, only a 5% residual activity of sulfamidase. So it's still present. However, it mimics MPS IIIA quite well because the protein is still present in MPS IIIA, but it's not working nearly as well as it should be to be digesting all of heparan sulfate and as well taking care of everything else the cell needs taken care of by the lysosome. So what we do is we isolate the brains from these mice of both MPS IIIA and wild type mice. We perform mechanical disassociation of the brain tissue. This takes hours, so long, but and you have to be very gentle because you subsequently kill all the neurons in every other cell type aside from some immune cells. So upon mechanical disassociation to extract live microglia, you're labeling via CD11B and CD45. These are two immune cell markers specific for, in this case, specific for microglia. So we know that anything that's CD45, CD11B high in our fluorescence activated cell sorting will be microglia. And you could actually see, I don't have it on here, but you can see that the microglia of the MPS IIIA model are actually higher in both CD11B and CD45. So the peak, this is probably a wild type microglia subpop. And normally you'd see a more activated, higher CD11B, higher CD45 population from MPS IIIA microglia. So then subsequently we can isolate RNA out of these cells and we can try and get an idea of what transcriptomically is happening in the context of the disease setting compared to wild type. So if you can see at the top right here, yes. What I'm discussing earlier when I was saying an enhancer, I'm sure most of you know this, but I'd like to break it down a little bit. A transcription factor will bind to enhancer, and typically this whole region will fold over and be here and close in proximity and work together with this promoter and the transcription factors binding here to enhance the target gene expression. So right now, what we're looking at is the mRNA that is expressed from these target genes. So via RNA-seq, we can pull out the regions of RNA that are poly-A rich via oligo-DT beads. So you know you're not getting any microRNAs, you're not getting any long-arm coding RNAs, you're just getting mRNAs. So it cleans up what you're getting. And subsequently, when we isolated these out, you're able to see, now when we took a broader look at gene expression, we can see that in a disease setting from a 3-week-old mouse, which is, oh, let me go back. 3-week-old mouse in P21, comparing wild type and mutant or MPS IIIA, you're seeing a drastic increase in pro-inflammatory markers such as IL-1 beta, TNF alpha, a downregulation of homeostatic microglial markers as CX3CR1 and TMEM119. And you're also seeing a drastic increase, and sorry, this is P240, the age of the older mice that we're comparing them to, the 3-week-old to the P240. And you're seeing a drastic increase in the damage associated microglial or macrophage phenotype genes. So ApoE, TREM2, TYROBP, ITGAX. And you're also seeing a drastic increase in the expression of genes correlating to the lysosome or phagocytosis, such as ALDOA, CTSD, AXL, and CD22. Now, another thing that we wanted to look at was, and this is because when you isolate out RNA from a population of cells that you've acquired via FACS or via another process, you're not necessarily getting a good idea of the subpopulations of the cells that make up the population that you isolated RNA from. So there could be subpopulations of the microglia, even in the disease context, where a small subset of the microglia are driving a large majority of the disease process or of the inflammatory signature that we're seeing in the microglial population, when in reality, it might only be, let's say, 10 or 15% of the cells that are driving this. So what you can do, which is a really cool method, is single cell RNA sequencing. So you're able to barcode individual cells via, you have beads, and you're including those beads, which have their own specific barcodes, into each cell via oil droplets. So each one of these dots signifies the RNA of one different cell. So these are each cells, and what you're seeing is SGSH signifying the MPS3A mice versus control mice. So we wanted to know if the entire population of microglia in each, the wild type and the MPS3A model, are all synonymous, or if there's different subpopulations that are more inflammatory or less inflammatory, or if they express genes that are specific to that subpopulation or not. So what we're able to see is that they separate quite nicely, actually. The green, or the blue rather, being the control or wild type microglia, and the orange being the MPS3A microglia. So you're seeing that there is a drastic increase of expression of genes like we were talking about before: ApoE, TREM2, TYRO-BP, SPP1. These are all genes associated with inflammation and neurodegeneration and the damage-associated microglial phenotype. So you are seeing a general increase in these genes on a whole. And you're also seeing, and as you can see from here as well, you're seeing an increase in ApoE, for example, on the subpopulation of MPS3A microglia. And you're also seeing an increase in lysosomal storage, or lysosomal genes, such as LYS2, CTSD, IGF-1, and then you're seeing a downregulation. Oh, let me go back. A downregulation in microglial homeostatic markers or identity markers, such as SAL1 or CX3CR1. And the important thing to consider too is that while there are different subpopulations. So you can see from, for example, like 1 and 2 here, this being 0, 1, 2, that 0 is almost exclusively, not almost exclusively, but a majority control or wild type microglia making up the RNA profile. However, you are seeing some MPS3A microglia that mimic the signature of this population as well. So while certain subpopulations, so this, for example, is 2, is almost exclusively wild type, right? So we are MPS3A rather. And when you're looking at the genes that are expressed in this subtype right here too, it's very strongly for ApoE for these damage-associated genes, and for LYZ, for example, for the lysosomal genes. So while you are seeing the several subpopulations of these microglia in both wild type and MPS3A, you're tending to see specific subpopulations kind of driving the disease phenotype as we're seeing. And we're still looking into why these specific microglia are expressing this. And what it could be is there's specific localization of these microglia or cells within areas where the environment is driving this. So there could be more protein aggregation, more heparan sulfate extracellularly, more cytokines, more inflammation present in that area. And that might be why we're seeing a trend towards inflammation and dysfunction in those cell types, in that subset of those cell types in the disease context. So the next thing that we did is that we looked at, well, so there's two main goals of this project, I'll say. The first main goal was looking at transcriptomically, right, which is what I just showed you guys, to understand whether the MPSIIIA microglia actually have that damage-associated phenotype, which they do. So now we're trying to understand specifically what regions or what transcription factor family members are driving this, or are driving the gene expression that's leading to this phenotype. So what our lab developed is a way to isolate out the nuclei, the cell-specific nuclei from different cells within the brain. This is very new technology. You're able to snap freeze brains, and you're able to homogenize this tissue after it's frozen in a fixative, so that the transcription factors are still bound to the DNA of interest. And using different nuclear markers and transcription factors, fluorescent markers, you're able to isolate out these. And what's cool is DAPI is usually a marker in cell sorting to signify like dead cells. So you wouldn't want DAPI positive, but when you're isolating out nuclei, you use DAPI to take in that's how you're isolating out the clean nuclei from the rest of the debris that is present in the processing. So the DAPI positive, like let's say nu-N, so nu-N is a marker for neurons, or PU1 or SPI1 is a marker for microglia. So, and as well OLIG2. So oligodendrocytes, microglia, and neurons are the three main cell types. We've also done for astrocytes and different types of neurons, so excitatory, inhibitory neurons, things like that. But what's cool is that we're also very proficient and excel at chromatin immunoprecipitations. So these assays let you look at what regions of the DNA, and you can either do like histone modification chip or a transcription factor chip. So you can either look at which cell type or where regions of chromatin are bound or modified on the histones, and what genes those are linked to, right? So, and this is actually much cleaner than doing chips on whole cells, because you're able to fix these nuclei in their frozen state. And a lot of times once a cell is dying or thawing or dying, you'll lose the transcription factor. So you won't be able to do a great chip. So if you can freeze immediately and fix your tissue in formaldehyde, for example, or DSG, when you're able to process these, you have a much better chance of getting a great chip. So we're able to look at, and if you can see in the top right, this is where a transcription factor would be binding in an enhancer region. So an H2K27 acetyl, for example, will give you the signal of active or poised enhancer regions, right? So, and this is specific to microglia. So SPI1 is what we were discussing. So you'd see a peak specifically for microglia in SPI1 or PU1. You wouldn't see it in oligodendrocytes or neurons. So this is a validation that our isolation assays, as well as our chip assays, are actually working and giving us the information we need to understand the cell type specific chromatin modifications that are occurring not only in the disease context, but also just in normal cellular processes. So RBFOX3 being nu-N, or a neuronal specific marker, OLIG2 being a oligodendrocyte specific marker. And again, this is a validation. So upon comparing the differentially acetylated, or for in H2K27 acetyl chromatin regions in microglia nuclei from MPSIIIA mice and wild type mice, we see a drastic chromatin reprogramming at enhancer regions as well as promoter regions, where MITF, right here, MITF and TFE family members are binding. So when I say MITF, it can also be TFEB, TFEC, TFE3, and MITF. These are all the family members associated. These are specific to different cells. They're specific to different cellular processes, but they all bind to a very similar region. It's the CAC-GTG, flanked by, and this is CA. You can see there's error. It's possibly a C or a T. So it's CAT or C-GTG. So this is called a clear region, which we'll talk about again a little later. It's going to come up, but that's what's so important to consider is that, and it's also flanked by T and A, that these clear regions, or CAC, GTG, are linked to these lysosomal genes, these other processes, these autophagic processes, and mTOR pathways as well, that we're able to link in lysosomal storage disorders, which was not able to be linked beforehand. So, and these came from these upregulated regions over here, the 695 upregulated acetylated regions present in MPSIIIA. So now that we know there is a connection between MITF and TFE and lysosomal storage disorders, let's get a better understanding of the way in which these processes are actually regulated. So TFEB, a family member of MITF and TFE, it actually translocates to the nucleus of the cell from the cytosol where it's bound to 1433, this subunit. When it's phosphorylated, bound to this 1433 subunit, it stays in the cytosol, it's not active, but upon inactivation of mTOR or upon efflux of calcium from the lysosomes, as well as from other processes such as nutrient deprivation or starvation, which would activate autophagy, and lysosomal stress, as we were discussing with heparan sulfate buildup, different macromolecular subunit buildup, and cellular differentiation. So in the context of cancer, for example, you'll see this inactivation of mTOR or this efflux of calcium and these other processes that would affect this, and subsequent dephosphorylation of TFE, B, C, MITF, TFE3, whatever factor, subsequent translocation to the nucleus where it's binding to these clear regions that are associated to the genes that we were talking about earlier. So this translocation, subsequent binding, and subsequent gene expression alterations or overexpression would lead to an increase in autophagy. These are the autophagy related processes, lysosomal function or overexpression or related to these processes, as well as mTOR C1 activity related to cancers. So the reason I'm talking about this so much is that it is a target for novel therapeutics, is that you can use an adenovirus-mediated TFEB overexpression. And this was actually done in the past, but an adenovirus-mediated TFEB overexpression had neuroprotective effect in glial differentiation from the MPSIIIA mouse model. Now, it's hard because an overexpression of TFEB would not necessarily have great effects either. So as we said, an overexpression of TFEB can lead to these processes. Sure, an overexpression of lysosomes and these things, you're able to possibly process these accumulations better of different subunits, macromolecular subunits, and different basically like garbage that needs to be processed. But this will also lead to inflammation, neuroinflammation, things like that. So the possibility of some type of knockdown of TFEB to limit this expression, or some type of like reversible mTOR signaling, because mTOR you want for certain processes, but in this case, if it is activated, it will stop the translocation of TFEB to the subsequent enhancers of interest and downstream gene expression. So, and this one I was talking about, decreased the intracellular glucosaminoglycans, decreased the size and number of enlarged lysosomes, and recovered morphology. So another thing that this was performed by Nathan Spann a scientist, great scientist, senior scientist in the glass lab, master of chip. If you ever need chip done, talk to him. He's the best at it. So is gain of TFEB at the enhancer of TREM2 of chloroquine treated macrophages. So basically, upon treatment, and these are in BV2, so this is a model of microglia cell line. So upon treatment of these cells with chloroquine, Nathan did an MITF as well as a TFEB chip-seq for these different factors, these different transcription factors. And you're able to see that upon treatment with chloroquine, which is a lysosomal stressor, you're gaining a peak right here. I don't know if you can see this right here between the no treatment and the chloroquine. And this is in the TREM2. So TREM2 is actually, has been linked to, in recent studies, upon its dysfunction or genetic variance or overexpression to late onset Alzheimer's disease. It's actually needed for microglia to completely enter into the damage-associated microglial phenotype. It's one of the factors necessary. It's not the factor, but it's one of. So as you can see, in the case of lysosomal stress, which TREM2 is, as I was saying, linked to Alzheimer's disease, other neurodegenerative diseases, microglial activated phenotype, you're gaining a TFEB site, a TFEB binding in this context. This leading to pro-inflammatory cytokines, inflammasome regulation, lysosomal biogenesis, autophagy, lipid metabolism, etc. So in summary, MPSIIIA mice are likely to be useful in dissecting the lysosomal role in acquisition of the damage-associated phenotype and link to subsequent neurodegeneration or vice versa. And we're able to see how the role of microglia, the role of lysosomes, specifically in the context of autophagy, of faulty autophagy, of these MITF, TFE transcription factors can actually affect neurodegeneration in different contexts. Now, microglia isolated from MPSIIIA mice exhibit early and prominent changes in gene expression that overlap with the DAM phenotype observed in mouse models and human of Alzheimer's disease. So what's very unique about MPSIIIA, and especially the model we use, is that we're able to see a disease phenotype, like I was saying, even in 3 weeks of age, versus other models, you have to wait years. And you might not even see the same drastic damage-associated phenotype. You won't see all the symptoms because we're not able to mimic it properly. We only give amyloid beta, phosphorylated tau, things like that, alpha-synuclein in the case of Parkinson's disease. And that epigenetic changes support roles of MITF, TFE transcription factors in driving transcriptional changes that we are seeing in the overexpression of autophagic processes, the overexpression of genes related to lysosomes, the overexpression of genes related to inflammatory responses, neuroinflammation, microglial phenotypes associated with damage, things like that. So moving forward, we're actually targeting the MIT, the team that I was working with at the Glass Lab will be targeting the MITF, TFE transcription factor family for stimulation or knockdown. We're trying to see the differences, what's going to be happening and what's a possible therapy for either MPSIIIA or potentially other neurodegenerative diseases, as well as translation into age-related neurodegenerative diseases. So the P301S mouse model is a hyperphosphorylated tau model, I believe. And sorry, this one was the first one's in collaboration with Yuming Li at Memorial Sloan Kettering. And this is the second one is with David Holtzman. So this is trying to see if in a mouse model that's not necessarily mimicking everything to do with Alzheimer's disease, for example, it is still an age-related neurodegenerative disease. And if this is able to have some therapeutic benefit, it can be expanded into other diseases and other treatments, as well as the role of IFIT1 IFIT-M3 in microglial activation. Now, this is it's a viral acceptance and transporter process. Basically, it's like a trigger for the immune system. So like I was saying, when heparan sulfate binds to the TLR4 pathway, for example, and this is driving immune function, but there's really not an infection. So you're getting a buildup of, not a buildup rather, but a overexpression of these inflammatory cytokines and overexpression of neuroinflammatory genes. There might be, if you can disconnect these because there's not necessarily an infection and try and suppress the immune system a little bit of that cell, then this might have beneficial therapeutic possibilities. So I just want to say first off, and this was a majority of the work that I did in the glass lab. I know we worked on HIV quite a bit too, but that's a whole nother discussion that I hope I can give in another talk. But for today, this is pretty much all I'm going to cover. I can briefly discuss with the work I'm doing in Guilherme Santos's lab with Natalia, the amazing grad student. And that is looking at the effects of lipids from a high-fat diet and a normal diet, and as well as the lipids extracted from the feces of animals treated with that high-fat or normal diet, and then looking at their effect on PPAR signaling and NF-kappa B pathways and different things like that, and the effects of different positively and negatively charged long-chain fatty acids, and seeing how they're either acting as an agonist, antagonist to PPAR, and subsequently changing lipid metabolism, and if that can potentially be a therapeutic for metabolic disorders. And as well, looking into the catalytic activity or the copper reductase activity of different histones that we're working on currently. So I first off want to thank, of course, members of UNB in the pharmacology department, Guilherme, Francisco, and Natalia for having me and being so gracious and nice and helpful and letting me come for only 2.5 months and get right into it and help out in the lab and try and share some expertise that I have and learn a lot as well. And I want to thank the people at UCSD that weren't in the glass lab, so Philip Gortz and Jeff Esco, for a lot of their work and support. And that was mainly with the production of the MPSIIIA model and their guidance in how to address a lysosomal storage disorder, because they've done many years of research with lysosomal storage disorders. And mainly Chris Glass and Johannes Schlahetsky. They have guided me greatly through my years in the lab, and I've learned so much and gained such a love for epigenetics, transcriptomics, and macrophages in general, as well as Hunter, who is an amazing scientist, an amazing MD PhD student, and just recently graduated, like I said. Can't say enough good things about him. Nathan Spann, a wizard of chip, a wizard of science in general, the smartest man I know. Martina, who's, oh man, the nicest, best technician that I've ever worked with, and a master of fax and fan sorting. Chris Ballack, a new grad student who has a ton of potential and is now helping and took over my help, who took over my part in the MITF TFE MPSIIIA project, as well as Rick Ziyang and Oh Young and Yi Zhao. They've all been great bioinformatically, and they've done a lot of the heavy lifting behind the scenes of the bioinformatics that you guys got to see in all those nice figures that were produced. So I want to say thank you so much. And if there's any questions or comments, please feel free to ask them now.
So thank you very much, Addison, for this nice and intriguing lecture, at least for me, because I'm not at this area. So there are some questions, but as I say, as we have the microphone, we do ours first. So
yeah, sure, of course,
not democratic at all.
Yeah.
I was thinking about this lysosome disorder and trying to, to figure out how do the MHC1 expression at the membrane of these cells with the disorders, how this occurs, and are those cells become a target to NK cells, for example, because they sensing cells with low MHC1 class 1 molecules in their membranes. Do you know something like that? I'm just asking you because I gave this class this morning to my basic immunology students, so I'm trying to figure out how it works.
Let me try. I think I understand your question, but you're saying specifically why microglia, or are you talking about NK
any
cells?
cell? If you can see, you can thinking about any cell that has these lysosome disorders,
right,
that impairs, I think, the production of peptides that will be charged in MHC class 1 molecule.
Right.
And if you don't have this, MHC, MHC2, any one of them, if you don't have this, you don't have the molecules at the surface of the cell. So you mark the cells, for example, for NK action, for example.
Right, exactly. So that's a great point, because in our gene expression, which I didn't put in here, but one of the main pathways that's affected is antigen presentation, like all the genes that are in this process and involved in that process and pathway. So it is greatly affected because the lysosome, like you said, is so important in that process, because you're breaking down pathogens and then presenting that antigen on the surface. So if you're not able to do that, you can't really distinguish between an actual infection, like with a virus or with some other type of pathogen, versus itself, right? So that is a major process of why this is getting so bad in the disease context.
I know you said polysaccharides, but I was wondering about this as a turnover process into the cell at all. Right. So I think it will make a mess, in the
Absolutely.
cell.
Yeah. And that's why you see so much peripheral and also CNS just inflammation in general, because it's every cell is thinking that they're being attacked essentially, and they're not able to properly present these antigens and deal with these issues. So it thinks that there's some type of global infection when really there's not. It's its own process. So that's why we're thinking.
Microglia, if you think about nervous system, central nervous system, you have this effect there.
Right.
Yes.
Say that again.
Yes. Even in the microglia, because you are in the central nervous system. So, but even there, you can see this effect of huge inflammation and cell destruction, for example.
Yeah, that's mainly what we were focusing on. This is all in the CNS. So we know that there's been other studies in the periphery. So people have been able to study immune cells and for example, in the heart and in the liver. I think I had that figure before, but and that's been studied in the past, but no one's really looked at. And people have looked at microglia in the context of lysosomal storage disorders, but not this heavily into the transcriptomics and epigenetics of it. Which is giving us a lot of information as far as like what's actually going on, what processes are changing, like you said, with MHC with class 1 presentation, and as well the mTOR pathway and things like that we wouldn't necessarily have, because in the past it was just looking at basic fluorescence imaging to look at like specific, we're seeing a lot more lysosomes, we're seeing a lot more aberrant protein aggregation, but now we're able to look at exactly what pathways, exactly what subtypes of those cells, where they are, what's going on in the environment that's driving that, and subsequently how we can treat that. So because now we know at least what the pathway is that potentially could be driving this. So, yeah.
So Guilherme, do you want to ask one question to Addison?
Yes. No, Addison. Well, first, thank you for the acknowledgement. It's really great to have you here again. I mean, it's fantastic. And always when I see your talk come up, many questions. So I have some couple of questions, but I will not bother you with all of them. Then we can discuss later, but just one that may be going to be of general interest.
Yeah.
In the beginning of your talk, you mentioned about, having this model that can, in the mice model, that can mimic this Sanfilippo syndrome in humans disorder. So what's the first question? What's the phenotype outcome of this disorder? How the patient looks like?
So the actual clinical manifestation or phenotype that you see is a child that, and typically, I should have described this. I thought I had it in there, but essentially the children will have, people will think that they have autism or things like that in the early stages around 1 or 2 years, but eventually you'll see skull deformation, and it affects the bones. So you'll see like osteoporosis at such a young age, and also like hyperactivity and craniofacial differences too, and also heart malfunction, liver malfunction, and things like that. So these children, typically you start seeing symptoms, and it's not very clear or clinically diagnosed in the first few years, but around the age, I mean, most people don't live past 15 to 20 years old with this disease.
Google it.
Right.
While
yeah.
I'm giving the talk, I Google it. I saw the image of the child.
Yeah. So
I was very curious about that.
Yeah. Yeah, exactly. So it's a very severe disease. So that's one of the reasons it's a good model to study other neurodegenerative diseases, because it's so rapid. And you, like, for example, with Alzheimer's disease, you're probably not seeing symptoms until you're, I mean, depending, unless it's early onset, right? But typically it's later onset. So you're seeing, in 60s, 70s, 80s, versus the first few years of someone's life, you're already seeing symptoms. So that's why in the first 3 weeks of that mouse model, you're already seeing symptoms. And I think your other question was,
No, this answer perfectly. So the other question, I mean, connect to this question is, this point mutation can mimic the human, I mean, Sanfilippo disorder in terms of phenotype?
Yeah.
Or so just the neurological side?
Right. So the point mutation, no, it's the whole periphery, everything, because that is the cause of the actual disease, is this one mutation. So that's why people have tried gene therapy in the past, and also I couldn't find the word when I was talking, but it's like protein therapy, essentially, or protein replacement of the SGS atriosulfonamidase gene, or not gene, but the
type protein. Is that you deliver the white type protein to the cells?
Exactly. So then, if you have that protein, you're able to break down heparan sulfate and hopefully start functioning better. Because the problem is not the lysosomes. The problem is the inability to digest this, because of the inability of not having this. So it should seem like an easy solution but a lot of the time it's really difficult too, because accessing the central nervous system, how do you do that, right, to treat this disease? And if it was in the periphery, it was exclusively in the periphery, you might be able to. But that was one of the problems we were having, as far as if you're trying to mimic this in vivo, you'd have to figure out some type of pump, some type of something to access the CNS to deliver this. If you're going to treat with a TFEB or something, some type of transcription factor. So that's part of the reason, but it is in the whole periphery, and in the mouse model as well. You see it in the periphery.
So talking about model, we have a question from our audience. It's a student of us. He's asking if is there a cell model or animal and mouse model is better than cell model? Can you have a cell model just to do your experiments in vitro or ex vivo?
I mean, you could, right? You could do, if you have cells, you can do some type of CRISPR SNP, essentially, of just and you can target that specific SGSH gene. You can do some type of knockdown or knockout of a cell type in a mouse model, and then subsequently have that. But you really want to do in vivo to look at some of these diseases, because if you're trying to look at why this disease is happening, what the process is happening, you can't get too much. And the other thing is, our lab was like very pivotal in researching the differences in microglia in a dish and microglia in vivo. So the whole when you put cells in a dish, you take it out from an in vivo model and put it in a dish, the whole epigenetic transcriptomic, but mostly epigenetic landscape changes completely within a few days, or with 21 days, things like that, a few months, completely different. So it's not even really a microglia. It's like you call it like a in vitro microglia now. It's not even really like a microglia. That's like BV2s, for example. While they are a good model to study microglia, they're not necessarily an in vivo microglia. So the best thing you can do is find a mouse model or find some type of model, preferably humans, right? Like if you're able to get that tissue, that's the perfect thing you can do. But in most contexts, you're not allowed, or you're not allowed, but it's very difficult to access those, especially brain tissue, right? Because that has to be after someone's passed away. Yeah, preferably in vivo for a disease like this, to try and study the whole process, cellular processes, environment, seeing how everything's affected. Yeah.
And thinking about this, you have an anatomy of the disease, like it begins
right.
some parts of the brain, and then it will, grow for the other parts of the brain. Is that correct to imagine, like something in time? You begin in a little part, and then it will expand. Is that correct? Am I thinking correct about this?
You're saying in vivo in the context of this disease, if like one part.
Yeah, in your mother, or for children, for example, I don't know. You have an influence of the environment, like
absolutely. Yeah, absolutely. That's why, like when we did single cell RNA sequencing, we're trying to see if there's specific subsets of the microglia. And that's the other thing, is when you're in a dish, typically microglia or a specific cell, they'll all look the same because the environment is the same. Like you're treating with the same factors to try and, let's say you're differentiating a induced pluripotent stem cell into a neuron, for example. You're giving it the same factors, you're giving it the same media, it's going to be the same. But in the context of a brain, based on the environment you're in, you could look different. Like neurons could look different in certain areas, and then specifically microglia could look different in certain areas, while they still have the same DNA. Their epigenetics, their transcriptomics will be different based on their environment. So that's part of the reason we wanted to do that. We favor in vivo models if possible. Yeah, but when you're doing a ChIP-seq, for example, like you're trying to analyze a ChIP-seq, or try and not analyze, but optimize a ChIP-seq rather, it's more about where the protein's binding to the genes of interest or enhancers of interest. And then you can use that in an in vivo model. So that's why you would do something like that in vitro versus doing something like this that's already been optimized into in vivo. Yeah.
Guilherme, quer fazer a próxima pergunta da nossa plateia?
Yes, no, but first I go for one from mine, because it's
you got the
sorry,
microphone.
Yeah.
No
Sorry guys, but I'm tempted to do that. One of the things that we did a lot of work was on H tags. So we show some data. I think it's a common marker to see the activation, genes activation, some regions, the acetylation regions. So first question is there any treatment for, what kind of treatment they do for this disorder nowadays? The second question following this question is, they are using or trying, are they trying, or it's correctly using H-tag inhibitors for this disease as a way to also to inhibit the inflammatory pathway?
So not that I know of. What I know is that, which I'm sure if you guys Googled it when I was giving the lecture, you saw that they've done AAV-mediated gene therapy for this. It wasn't as successful as they would have hoped. They've also tried protein therapy, protein replacement therapy. But like I was saying, there's no real treatment for it currently. And what Philip Gortz was using different breast milk sugars to try and have like an anti-inflammatory property, but it wasn't working as well as they would have hoped. So that's what that lab, the Gortz lab, is kind of looking at, is trying to use and find different therapies to try and treat for, because no one really right now has a therapy for it. So that's why we're trying to figure out if we can, instead of affecting or going for gene therapy, having some more like accessible approach or easily implemented approach that has less risk than like gene therapy would, for example. So, but no, currently not that I know of. And I also don't think that I don't know. I haven't looked into it if they're using HDAC inhibitors, but not that I know of. Yeah.
No. Okay. Thank you. So
Yeah.
I'm going to go for a question from the great students that are seeing that. I think it is a quite nice question as well. So Monica Garcia is asking, astrocytes are important also to activate microglia. So do you have any data involving astrocytes in your studies? I don't think that you have, but can you guess what would happen? Or I mean,
Sure.
Have you figured out about this?
Yeah, so we have looked at astrocytes a little bit. And while they are important for activating, microglia in this disease context are kind of driving themselves a little bit. But yeah, they are important. And while it's also difficult to isolate astrocytes from the specific model that we had for isolating it, we have it optimized for these 3 cell types. And when you start increasing cell types, it becomes more difficult. So while I do address the fact that astrocytes are important as well, I would say they're more important for, I guess in this disease context, we can look at how the environment is playing more of a role and the disease is playing more of a role rather than astrocytes in particular. So normally, yes, but I would say in this, there's much more happening in the environment that's driving this microglial activation rather than just astrocytes, which would normally be happening. So it's a good question though. Yeah.
So we have some questions about other neurodegenerative diseases, like Parkinson's disease, Down syndrome disease. Is there any idea of lysosome dysfunctions related to this disease too, or not?
Yeah. Definitely. That's why we were kind of looking at this, because our lab, or Johannes specifically, understood that there were lysosomal dysfunctions present in Alzheimer's disease and Parkinson's disease and Huntington's disease already. And that is partly manifested in the aberrant protein aggregation, like amyloid beta, hyperphosphorylated tau, things like that. Because normally you would be able to process these buildup of protein. So if there is too much for these lysosomes to be doing because of a buildup or some type of faulty autophagic processes that might be linked to the TFE, MITF transcription factor family members, then that could be the connection. And that's why we're trying to study this and use this as a model to study other neurodegenerative disorders, because most other neurodegenerative disorders also have lysosomal dysfunction as well.
Lysosomal.
Yeah.
Oh, very interesting. I didn't know that. Talking again about models, a student is asked if the organoid model would work. The organoid, not the cells, isolated cell, but organoids. Do you know anything about that?
Yeah, so we actually did some work with
they don't want to use animals. Do you know that?
Yeah, I could tell. They're like, let's find another way. Organoids are so complicated. They're very difficult. I know that I think that someone in the pharmacology department does organoids as well. So I can definitely relate to how difficult they are, because we used to work with organoids. But, and what we would do is we would generate organoids and we would implement microglia into these organoids. But what the problem is that if you're building a neuronal organoid model and trying to implement microglia into that, it's hard. You don't have astrocytes. You can't really manifest the same types of problems, and you can't simulate the environment of an actual brain. And the other thing is, as you're getting an organoid big enough, a neuronal organoid at least, people have been able to develop organoids with vasculature and have different blood vessels that can deliver nutrients to the inside of these organoids, but it's very hard to do that. So you either focus on vascularizing an organoid, or you focus on having a smaller one with microglia and then trying to mimic that. So it's the technology right now. I know there's some people doing amazing organoid work, but it's not where it needs to be to properly study in a cheap way. And not that mice are cheap, but it's at least more of a surefire way, because sometimes we had a lot of difficulties with organoids in the past, and don't model microglia the same way in an in vivo model, but in the future, for sure, I think that would be a great way to study it. Yeah.
I think the main idea to the students, maybe we didn't find the way to get these diseases all set it in these organoids, because we got a seminar last year here from a guy that uses brain organoids to study Zika virus microcephaly.
Yeah.
So, and this guy was very, very, successful with his model, but it's quite different. It's quite different from what you are talking about, an organelle inside the cell, a very specific type of cell, and that suffers with the environment of the other cells there. So I think it's quite different when you think about this. So
what we were actually doing, we worked with some people at Salk as well on this project, because what our lab focuses on in big part, and like I was saying, you can use an organoid to study these different diseases, and it's for sure better than having like a dish of microglia, because you need to have cells talking to each other. So I totally agree with the model, but what we would do is we would implant the organoid into a mouse brain. So we would carve out part of a mouse brain and implant that organoid with microglia in it and try to see the differences. And while that's unpublished data, we saw differences between the microglia in an organoid and microglia in an organoid that are implanted into a brain. So no matter what, in vivo is the way to go. But I agree that and that's only if you're trying to study like the epigenetics and transcriptomics and behavior.
That complicates a lot.
Right. But like, if you're trying to study pathways and things like that and do ChIP-seq assays and try and do imaging and other things and look at, for example, like Zika virus and how that's affecting, let's say, I don't know, cellular functions, that's fine. But, for this one, and it's also in part because of the history of our lab's research, that we know that's if we're looking at what we're trying to look at, the best way to do it is this way. But I agree that once organoid methodologies get more advanced, it's going to be the future for sure, because it's just easier than raising animals, and it's more ethical. I think
it will be human cells, and maybe you can give. Right. You can have different contests, maybe. I'm just thinking.
Exactly. So totally. And then what I was I actually gave a talk in Guilherme's pharmacology class on a project that we had worked on in collaboration with other people at Salk, but you're able to take fibroblasts from patients and then de-differentiate those and redifferentiate them into neurons. So you're making induced pluripotent stem cells, and then from fibroblasts directly into neurons, or you can even turn fibroblasts directly into neurons, and then you can make organoids from that. So it's definitely the future. I agree. Yeah.
And how about I think it's the last question, at least I have seen here. How about non-coding mRNAs? What are their roles in this kind of disease? Like non-coding RNAs, miRNAs?
Yeah. So we haven't looked too much into that, honestly, but and I think, I can't really answer that question because it's not something we
can revisit your transcriptomics, try to find them, because probably you will find them there.
Yeah,
you think?
Most likely. And that's just a different methodology that we're not that well versed in, to isolate that.
The data is already with you. Don't you think?
Exactly. It's already there. But for right now, not to my knowledge, I'm not aware of any relationship or
actually, sort of. That I don't think that you have the sample yet, because you isolated mRNA. It's a poly-A tag.
Poly-A.
So
they contaminate them. We did this. Okay. And we found some. some. Very, very interesting. So you can find it, but I think if you see your transcriptomics data, you will find them.
I'll look in there. I'll write it down and I'll check, and I'll get back to that question. I'll ask. I'll answer that later. Yeah.
So Guilherme, do you have any questions? Any more questions?
No, I have a question following the discussion about organoids and the best model. I think it is a very tricky question that John Alves did. And I think it is always a problem. But so he asked, does taking the cells out of the context, mainly the extracellular matrix context, could have any effects in the results? Is there a way to control or measure this effect? This man, he's the reviewer of your paper. He's being the lawyer. So yeah, it's a well.
We discussed that a lot. Of course, the in vivo model is the best one, of course.
Yeah.
We cannot do this in humans. At least at this disorder, you can have this model with 1 point mutation.
Right.
But when I Googled it, because I did, I have to confess, when I Googled it, I saw that it is a recessive disorder. So you have to have the both alleles, the both chromosomes with the point mutation.
Right.
Is that correct? So you don't have heterozygosis on that. So
right, because it's one of those. It's like if you have one allele with, in this context, like with the affective gene, then you're able to code for sulfamidase. So then it's okay. And it's I don't think it's, what is that, haplo insufficient? It's haplo sufficient, so it's fine with just one. But yeah.
Okay. No, but back to the question, I think he wants to see if these results are biased. So one thing that I think is relevant to emphasize, that when they take the cells, they go for flash freezing, and they start and then fix the cells. Mm-hmm. So at the same time, all the process is frozen. Yeah. So that would be a sort of to control. I mean, the cells that is out of the context in terms of matrix and all the communications. So Addison, if you want to say something.
Yeah, I didn't know if that was a question or a comment before, so I'm sorry. But yeah, so the reason that we do that is, like I was saying before, when you're isolating out live microglia, typically it takes hours. And then sometimes even if after doing fluorescence activated cell sorting, it's been 8 to 10 hours. And already in that time, you're seeing changes. So if you haven't fixed them yet, there are epigenetic modifications in that time, because now they're in a different environment. They're being thrashed around in a machine. They're in an inflammatory state even, versus when we isolate out, when you snap freeze a brain right away, because we can't isolate, we have tried, but it's very, very hard to isolate out RNA, like good mRNA from frozen tissue. But if we're looking at strictly epigenetics and trying to do chip, you get a very clear, clean histone mark chip, as well as transcription factor chips, because you're double cross-linking, you're single cross-linking, you're doing whatever, but everything is in place when you freeze, and then you're able to sort it out. And it's a much cleaner way of looking at, a much more effective way of looking at epigenetic modification. So I guess pertaining to the question is, when you take something out of its environment, I can't remember if it was a Science or Nature paper that Chris had published like 5 years or so ago, but that was mainly on the changes in microglia when you take them from in vivo to in vitro, and the changes that you'll end up seeing. So, yeah.
Okay. So it was very nice discussion. Put our brains to think about it. And thank you very much for the nice afternoon we spent together here.
Awesome. Yeah. Thank you so much. Yeah.
And hopefully you find some tournaments, jiu-jitsu tournaments, to come back and come back and come back again.
Yeah, absolutely.
Ok? Então, thank you very much. Muito obrigada, Guilherme, por estar aqui também. Obrigada mesmo.
Poxa, muito obrigado, André. Obrigado, Addison. It is really great. Again, I mean, André just mentioned that it's going to have plenty of tournaments of jiu-jitsu, so probably you're going to be back very soon. And also, there's some nice waves for him to catch. So this is it's very attractive. And nice science as well.
And science, don't forget science.
A little bit, just a taste.
Yeah. Yeah.
Okay, thank you, Ad. Thank you, André. Thank you, Givago, for all the organization. It's really great, very professional.
Our internet wizard. He is our internet wizard. So, gente, obrigada a todos. Pronto.